I'm Lisa Standley. I'm the curator of vascular plants at the NEBS Herbarium, and a longtime fan of the Cyperaceae and Carex in particular. So what we're going to do today, I'm going to talk a lot, but feel free to interrupt me anytime. Things like, why do we collect? The ethics of collecting, what our goals are, what you need for equipment, how we take labels and data collection, a lot of tips and techniques for how to deal with your plants and your specimens, and then we'll do some practice. I've brought in a couple of bags of plants and some presses, and we're actually going to try our hand at actually pressing some plants. So, talk a little bit first about herbarium specimens and why we collect herbarium specimens. And there's a whole host of reasons. Some people collect them just for their own identification purposes, so that they will have a record of what they've seen. It's often easier to identify a specimen when you've got it home and your microscope than if you're in the field, especially when it comes to grasses and sedges. But mostly the organized herbaria really function as a research tool, and research in plant systematics, the relationships and evolution of plants, plant ecology, plant community structure, plant community differentiation, and floristics. Where are plants distributed on the landscape? What's the flora of particular regions? And what all this lets us do is both do scientific research in these fields, but also really understanding change in the natural world. What has happened in the past? Can we predict what's going to happen with climate change? And it's really important that herbaria represent the diversity of taxa, all the taxa in a region, that they've got a range of morphological variation, so they show all the variations in morphologies, that we represent both the historic and the current flora and its distribution. And we also serve as a repository for voucher specimens. If people are doing ecological studies and collect specimens to document what was in their plot or their study area, we will take those specimens as well.
I have a question about voucher specimens. Is there a standard, is there a standard of what geographic area should have a voucher specimen? Like, is it towns or counties or what? Because I come across a lot of specimens, stamped voucher, but it doesn't say like voucher for where.
Well, it should, generally speaking, and we can talk about this with labels. If you collect a voucher, vouchers are usually associated with particular studies, like a flora of a certain town, or the effects of clear cutting at Hubbard Brook or something like that. And they should definitely say voucher associated with a particular study. If you're just collecting plants to document their existence, they don't have to say voucher on them. It's only voucher if they're associated with a real scientific study.
Okay.
But in terms of, I'll get to...
But like if it's like the first time somebody's documented.
So the New England Botanical Society's herbarium, which is known by the acronym NEBS from its past history, we actually have about 350,000 specimens, which is the largest collection of New England plants in existence. And we've been collecting so we've been amassing herbarium specimens that have been collected since the early 1800s. Our collections go back into the 1840s. And the collections really span what I would call the golden age of plant collecting, the 1880s through the 1930s into the 1940s. The majority of our specimens, I think, were collected before 1920. So it's a great, as I tell people, it's a great record of the turn of the century flora. Not the turn of the last century, but the turn of the century before that. And what we really don't have are good modern collections. People don't collect anymore. people haven't collected much since the 60s, except for a few people here and there. And that is a real problem in trying to assess modern plant distributions or modern plant phenologies. When does something flower now compared to when it flowered in 1880? Well, if you had a herbarium specimen from now, you would know. Do plants still occur where they did 100 years ago? Who knows? We don't have the data in the herbarium. So there's a real lack of modern collections. There's also what I call sort of collecting bias in what we've got in the herbarium. People like to collect ferns, and they like to collect rare plants and orchids. Those are actually overrepresented. There are some rare species for which we have more specimens than we do of common species, because people go after them. And there were lots of amateur collectors in the end of the 19th century, and they liked ferns. They collected lots of ferns. There's also a locality-based bias in collecting. The old-time collectors would collect in popular places, often vacation spots, places that were accessible by train before the age of cars. So we've got tons of collections from the White Mountains, or the Holyoke Range, or Mount Desert Island, places where people could get to and collect. Other locations, we don't have really much for those localities. So what we need to fill those geographic gaps. We need modern collections to show us modern distributions and modern phenologies. We need collections of introduced and invasive species to see where their distributions are, how their distributions are changing. And it's people like you, volunteer collectors, who are going to fill those gaps. In the 30s, 40s, even up through the 60s, there were academics who focused on New England, who collected like mad. But academics in botany these days don't do that. If they're doing field work, they're in the tropics. They're not doing much, if any, field work. They're really focus much more on genetic analyses. And they're not out collecting the mass numbers that they would have in the 40s. And it's really amateur botanists who are the source of most of our plant collections. So what I'm hoping to do is stimulate people to get out there and collect. And what I want this workshop to do is to help show you how to do it, the best way to do it. Yeah.
I'm going to talk about, I think a lot of people aren't physically collecting because they're iNaturalisting or
iNaturalist, the reason that we have herbarium specimens, and we'll talk about this when we talk about rare species, is that herbarium specimen records are forever. That plants that go into a herbarium are in a publicly accessible repository, accessible to everybody, and they will be there in 100 and 200 years. Electronic records, iNaturalist or other databases, we don't consider them permanent records. That who knows where in 2 centuries, what that kind of electronic data, where that's going to be, or how accessible it's going to be. Herbarium specimens are permanent. There's also the reason for a voucher, a herbarium specimen, is that you can verify, scientists can verify what it is. So many iNat records cannot be verified because people don't take decent photographs. They don't photograph the right characters. You know, if you're working with grasses and sedges for identification purposes, you have to dissect the flowers, look at the seeds inside the flowers, measure those seeds, and you can't do that on iNat. And so you come up with a pretty picture which can't be verified. So to be verifiable and permanent, you have to have an actual specimen. So one of the things I did when I first started as a curator was to figure out where we had geographic gaps. And there are really great ways to do that. This is a map from Ray Angelo and Dave Buford's New England Atlas. And they've mapped out, based on verified records, county distributions of all of the New England plants. And what I did was I went with those maps and lists of common species. And I looked for gaps where places, counties, at the county level, where common species had never been collected. And when I had 3 or 4 or 10 common species that all showed the same pattern, I knew I had a gap county. And we identified a lot of those gap counties. Interesting, some of these counties in central New Hampshire, Sullivan, Merrimack, Belknap, almost never collected. Northern Connecticut, a big gap. A couple of counties in Maine, a couple of counties in Vermont. And so we've been targeting those counties to try to build the records. There are also, we're always looking for new records of plants that have never been collected before at the state or county level. We are looking for town records as well. We don't have physical herbarium space to have a specimen for every species in every town. So I really focus at the county level. But it would be nice to have several species per county so that we've got a good record. We're looking for what I call underrepresented taxa, which are often are weeds. You'd be surprised at how few dandelion species there are in specimens in the collection. And even common things that are underrepresented, like, do you know pathrush, Juncus effusus? It's everywhere. 10 years ago, I collected it on Nantucket. It was the first herbarium record for Nantucket. I'm sure it's been there for a century. Nobody's ever collected it. So you find those new records all the time. And it's also important for conservation concern to have herbarium records that document populations of rare species that are uncommon. As I said, people report them through iNaturalist, they report them on the state heritage databases, but those aren't permanent records, they're not verifiable. And it does come up with a problem. I was asked to check a population of Carex typhina, which is a state-listed sedge, because there was a electronic record of it, but no voucher specimen. And there's another species that looks really similar to it. And you actually have to look at the plant to tell what it is. So if we'd had a voucher specimen, it would have been easy. I've done a couple of floristic studies of towns I've lived in and properties that I've worked on. And when I started, I didn't always take voucher specimens. And going back over my work now 10, 20 years later, I look at some of them and think, I wonder if that's really what that Dicanthelium species was, but I don't have a voucher. So it really is important. So next I want to talk about collecting ethics, which is a question that Vicki brought up earlier. So there are ethics associated with plant collecting that I hope people really learn. And the first is very similar to the basis of medical ethics, do no harm. When collecting, don't have a negative impact on the population viability, on any ecological processes, on aesthetics. So be very careful not to have negative impacts. You need to be respectful of property owners to obtain permits and permissions, and I'll talk more about that. You need to be aware of state laws and regulations concerning rare species. And to help you with all this, we actually have a guide to ethical collecting on our website. And I'll show you those websites later, but I also have a handout for you that lists all the websites. And the permits and permissions is really becoming a very important issue. There are many properties, state and federal, where plant collecting is prohibited by law. Not just discouraged, prohibited by law. And we'll talk about that. Back to the negative impacts. The classic example of the negative impacts of plant collecting. Do you know Franklinia altahama? The American tea tree. It's a gorgeous camellia family flowering shrub that the Bartrams collected in the early 1800s in Georgia. And they collected it, and they collected it, and they collected it, and it no longer exists in the wild. They wiped it out. They and other collectors completely wiped out the only population. So collecting ethics and negative impacts is something to really be aware of. In terms of collecting ethics and permits and permissions, many public lands, state and federal, things like state wildlife refuges, state parks, DCR properties, as well as national parks, national wildlife refuges, other national lands. It is prohibited by law. And because there is a legal prohibition, we as an institution at NEBS, we cannot accept a specimen that was collected illegally. We would be in possession of essentially stolen goods. So we can't accept something that was collected from those properties without the appropriate permissions. So we require people to put that information on their herbarium label. So I see one of our collectors did a botanical inventory of a DCR property in Fairhaven, Mass, under a permit, and all of her labels say collected under permit number such and such, as they should. It's really very important. It may not always be easy. The national parks don't like to give permission, and National Wildlife Refuge people don't understand. And you may not be able to actually find a person who can give you a permit, but at least try. But in those cases where you wouldn't be collecting if you didn't have a purpose. So you would know about it in advance, and you would coordinate and get the appropriate permits. This also applies to lands owned by private conservation organizations. Nature Conservancy, Trustees of Reservations, Mass Audubon, they're private land, but they're dedicated to conservation. If you're going to collect on one of their properties, you should ask permission. And note on your collecting label, collected with permission of. Eric?
I was just collecting on the Nature Conservancy site yesterday, permission, but I don't have a number, so what would I say in my label?
Collected with permission of. For that kind of thing, that's all you need to say. Only the state and federal agencies would issue a numbered permit. Same thing I collected on a group with land of the Kennebec Land Trust with their permissions, and so we say that on all of our plant labels. It's very important. And, it's also good to talk, if you're going to collect on private property, private lands, to talk to the property owner and get their permission to collect. Most people don't care, wouldn't know that you were even out there, but it's good to make sure that they are okay with your collecting a few plants. On our website, under the ethics page and permits, we have suggested language. Send an email to the property owner saying what you want to do and why it's important. It's a great way to educate them about the value of plants and the value of collecting as well.
So can you accept specimens from private land? If you accept a specimen from private land, do you have to then contact? Okay.
No. We don't have, for NEBC, we don't have a hard and fast rule about collecting with that kind of permission. We encourage you to do it. We don't require it. Okay. The only thing we require is where it's required by law.
Okay. I would imagine that you probably want to have like a purpose or be part of a study or, I mean, you're not going to get it seems unlikely that an amateur collector is just going to kind of be able to waltz in and say they're interested and they want to see what's there.
Well, you can look at what we've got for language, and one of the ways to approach them is if you're an amateur, is to say, you know, I'm an amateur botanist with the BCCCI, and we're interested in documenting the flora of Cape Cod. Would it be okay if we collected a specimen on your property? So very high level stuff, and that works just fine. And we don't require permits. As you know, the NEBS herbarium is housed at Harvard. We're co-housed and co-filed with the Harvard University Herbaria. They require written permission for every one of their specimens. We don't.
So run that, you're not requiring it for private property?
No, nope, only for,
Nice to have it.
it would be nice. We encourage you to do it. We think it's good ethical practice to get a permission, but we don't require it. And we understand that there are what we call opportunistic or accidental collections. You didn't intend to be collecting plants in the White Mountain National Forest, but you were on a hike with some friends and you just saw something that begged to be documented. Not something rare, but you saw an invasive where there shouldn't have been one, or a really great specimen of a dicanthelium or a juncus that you thought, this is really great, and we really should document this. Well, go ahead and collect it, but try to maybe ask permission afterwards. With regard to rare species, again, these ethical prohibitions are really important. And you really have to think about this because all the New England states have laws that require them to list rare species, rare plant species. Not all states have the same laws about protecting those rare species. Maine, for example, lists them, but there's no legal protection. You can collect any rare species in Maine anywhere you happen to want to, maybe not on Maine public land. The collecting laws vary a lot state to state. So you really, here where permits are required by law, you do need to get a rare species collection permit if you're going to collect rare species. And we do want to see that permit. We want to have it noted on your label. So you really need to know your state requirements. You need to know your state list of rare plants. And again, there might be some opportunistic or accidental collecting. So you're out collecting sedges. I always use sedges as my examples. And you collected something, brought it home to key it out, and you keyed it out, and it turned out to be a state listed species, which you couldn't have known in the field. You had to key it out. It was a state listed species. You collected it. We will accept that specimen without a permit, but I will have to call the state botanist and say, Bob, is this okay? So we have to get after the fact permission for that kind of accidental collecting. So when do you collect a rare species? There are certain standards, again, which are spelled out in on our website. Only if it's necessary to document a new population. If this is a new site for that rare plant that's never been documented before, let's get a voucher. If the last record from that population was 50 or 100 years ago, let's document that it's still there now. And we always recommend to, if you think you might want to do this, check in with your state botanist to see if a new specimen is really necessary.
The regs talk about it being 25 years.
Yes.
So I noticed sometimes they say 20, 25. Is the 30 a valid number?
We came up with 30 sort of as a consensus for from different states as to what they would like to see. So it might be 25 in some cases. It does. Yeah. But rare species records are really important, but you've got to be careful about when and where you collect.
And does that go for how far down do we go, like special concern, does that count?
So the question is, at what level of rare species does this apply? And it applies to those rare species that are legally protected. So in Massachusetts, special concern is legally protected. Watch list is not. So watch list is okay.
But all of these rules don't apply from, the collection rules in terms of rarity, even though they could be extremely rare in the state. That's something I'm working on right now. I'm going to try to get them protected by the state. But my mosses aren't protected by the laws.
No, they're not. They don't list mosses. So collecting ethics doesn't just apply to rare plants. It also applies to non-rare plants, not rare listed plants. And as well as rare, and I'll talk about those. None of this applies to invasives. Common species don't collect if there are fewer than 10 individuals in a population. So if you've got a very small population, don't collect it. Don't collect anything. For certain groups like the lilies that are very long-lived but grow very, very slowly from underground rhizomes and tubers, we recommend that you collect only the above ground parts. Don't collect the roots. Leave those to live again. For common species, how much do you collect? Well, you try to get at least one plant. If they're tiny, get more than one. You want to have a diversity, you've got space on your page, collect more than one. A herbarium sheet that has a single Drosera rotundifolia in the center of it looks really silly. Woody plants, just enough to fill that sheet. And one question that people sometimes ask is about duplicates. Collectors in the past would collect large numbers of specimens from every population they saw. Some of these collectors were selling their specimens, the old timers in the West. Some of them were just collecting and would distribute them among many herbaria so that every herbarium would have its own sheet. Now, since virtually all of our plant collections at all the major herbaria are online, you know what sheets are where. And not every herbarium needs its own vouchered specimen. So we tend to only collect now a single specimen. The only time we differ from that is sometimes for NEBC collecting trips. We'll do a joint NEBC trip with another institution. We did one last year with Brown University, and we collected duplicates so that Brown would have a specimen, we'd have a specimen. But generally, don't collect duplicates, just get one. One is actually enough. With rare species, if there's fewer than 10 individuals, don't collect it. If there's fewer than 100 individuals, you should probably only collect part of the plant, not the whole plant, not roots and all, just a piece of it that's good enough for identification. With rare species, we will accept good quality photographs. So if you can't collect a plant specimen, you could take a photograph instead. You can only collect a whole plant from a rare species if there's more than 100 individuals. And those could be ramets or genets, but only if there's a lot. So that's why probably some rare species populations go undocumented, because the populations are too small. But even if it's small-ish, you could collect a little piece of the inflorescence, just enough to document it. So let's take to how do you collect? What do you need to have with you to collect? And there's a certain amount of equipment that's handy to have. You need a notebook so that you can collect data. You need pens and pencils, and I always say multiple because I lose them in the field. I always like to have Sharpies with me for some data collection. It's good to have a pen knife, a small knife. It's, for woody plants, you've got to have a pair of clippers. And you need to have something to dig plants up. So there's several tools. There's the old-fashioned trowel. There's this handy little dandelion digger, which is also pretty helpful. But what I've come to like best, I think most people do, is a Japanese digging knife, which is called a hori-hori. And mine has gotten stuck. There we go. So it's a handy little serrated blade knife. This edge is sharp, this edge is sharp and serrated. And this is great, it's got a good full tang handle. And you can drive this into the ground, cut around a deeply rooted plant, and pull it up, which you can't do with a trowel. So this is a really, really handy garden tool. When I first started using them, you could only get them online through specialized garden shops. You can now get them anywhere, in any garden center. You just have to be careful using them, and I always use the sheath because they are lethal weapons. And in any major collecting trip, sooner or later, I nick my hand. But they are really, really handy things. It's also good, if you're doing a lot of collecting, to have a field press. And you don't want to carry this press in the field. You want to go in the field with something, with just being able to grab plants and collect plants and carry them out to another site. So there are options for field presses. The simplest is 2 of these lattice boards and some cardboard, and I bind mine up with a bungee cord. You can use a strap, and then that's something small, you can just carry it in the field. You can make your own with some cardboard or masonite and a lot of duct tape, and you've got a nice inside packet to put all your plant specimens. Herbarium Supply Company makes these really nifty field press, traveling field presses. And this is great. It's got a strap. It opens up. You can carry newspapers in it. You can carry cardboards, and you can load it up. I can put about 50 plants in here and compress them. This is a really handy tool. They cost about $60 or $70, but they're really, really great things to carry in the field, and they've got a pocket in the front where you can put your clippers and your knife and your pen and your notebooks. So very, very handy stuff. You want newspapers because you're going to put your plants initially into newspapers. If you've got a college newspaper or a local town newspaper that's substandard size, your herbarium sheet wants to be about 17 by 12. So I take the Globe and the Times and just cut them down to size. So you want a lot of newspaper, comics or not, that you can use. And then back at the lab, you want an actual plant press. And this is a very typical plant press. It's a sandwich of lattice boards, cardboard, and paper blotters. Some people don't use blotters, but I find them to be really handy. They help you press better as well as being absorbent. But that's your basic press. Okay, we're gonna come back to that. So, aside from you're out collecting, you've got your equipment, you know you've got a press, you know what you're collecting for plants, but one of the most important parts is data collection. That's why we have our notebook and our pencil, because specimens without data are worthless. I see a lot of historic collections back from the 1800s, often that simply have the name of the plant, the date, and the name of the town. That's pretty useless. I've actually been offered historic collections that I've rejected because that's at best, all the information we have. And it's not enough to really be useful. So what you want to write down, and you want to write this down in your notebook, not on the newspaper, in your notebook. The location data. Where did you find the plant? The county, the town. I like to know the distance from the major landmark, like the distance from the town center, the distance from near a road intersection, if you're on a trail, the distance from the trailhead. And you might not get that, you might not have that information in the field, you might add that later. But it's really important to also get the GPS data. So know where you got it in terms of GPS, know the elevation. And GPS these days is so easy. Everybody's got a cell phone. All cell phones will tell you the GPS location. You don't need a fancy GPS for this. And then you want ecological information on the plant's habitat. Where is it growing? What's the slope? What's the aspect? What's the substrate? What's the overall plant community? Can you list a couple of the canopy dominants? Can you talk about the shrub dominants and the herbaceous dominants that are growing with your plant? That, for ecological studies, is really important information. You might want to put in other notes just about the plant. If you've collected just a part of the plant, it would be good to know how tall was it? You know, was your plant 6 feet or was it 60 feet? For a tree, you don't collect all of a tree, you might want to know the diameter of that tree. It's very useful to write down things like flower color, because colors fade. Some colors stay good, surprisingly, like red for cardinal flowers stays vivid red for a century. But some blues and pinks and even yellows fade. So color of the flower is really important to note down if you've got something in bloom. And if it's got any odor, is it fragrant? Note that down too. If you see a pollinator on your flower, maybe note that. That could be helpful. And things like if it's annual or perennial would be good to also note. If you've ever tried to key Persicaria, the smartweeds, like the number 2 couplet in the key is perennial or annual. Well, if you've got a herbarium specimen that didn't happen to include all the roots, you'll never know. And you have to key it both ways, which is a real pain. So if you can tell if it's annual or perennial, write that down in your notes. Try to be helpful to yourself and other people. So that's a lot of data, and this is a very good plant locality, plant label that was generated from some of our work last year for work that we did in Cheshire County, New Hampshire, that talks about the location, the plant community, GPS coordinates, who collected it, the date, and collected with permission. So that's a really good, pretty complete plant label. And you've got to collect all that data in your field. Some people number their collections, which is, and this is optional, but it can be useful to you as a collector. Some people do it sequentially. They assign every specimen they collect a sequential number. And some of the big collectors of the golden age, like Arthur Stanley Pease, who collected all over the world, his collection numbers, and I'm still working on his collections, are up in the 46,000s for his lifetime collections. It's amazing. Other people do it by year. So they start the collection sequence anew every year and put the year either at the front or the end of that number sequence. So this is just an example of one of Dick Eaton's collections. Dick numbered his specimens sequentially. And this, by the way, is a lousy locality information. We know the county and we know the town. And of course, you know, there are hardly any roads in Pepperell, so it would be easy to find it again. That's a really lousy location data. Don't ever do that. So then, before you do anything else, you've got your information in your notebook. You write identifying information on your piece of newspaper so that your newspaper that you're going to put the plant in is labeled. And I like to do it sort of along the bottom edge of the newspaper. And and it's good to put your collector number on there so that you've got a cross-reference between your notebook and the specimen. So top one is an example of something with a sequential number. I didn't know what species of cyperus it was, so it's cyperus A, and I collected it on that date at a place called Round Pond. Or the other one has a year number as the identifier, and just the name of the species and the date. It's very helpful to always put what you think the plant is and the date, and some kind of annotation about locality. This one, I wasn't using a numbering system, it's just the date. It's some species of spike rush and where I collected it. And you will the reason you do this, of course, is someday you're going to type up your notes and make plant labels, and you've got to know which label goes with which specimen. And so you've got to have the data on your sheet that matches your notes. Okay, so you're ready to collect. Now a little bit back to the overall collection. What do you collect? So only collect plants that are spontaneous. That is, don't collect anything that was planted. We're only interested in things that are growing on their own volition, as it were, not planted. There's a few exceptions to that. I collected not long ago in a piece of conservation land a meadow sequoia, a dawn redwood. I didn't know if it had naturalized there or was planted there. So I collected it to document that we had it. I later found out through some conversations that it was probably planted. We have a little Johnny Appleseed who is running around planting dawn redwoods. But I think it's still an important record because what if, when that plant reaches maturity, it starts to reproduce and we get little dawn redwood seeding in? Won't it be nice to know where the parent plant was? So it's still useful. So when you collect, spontaneous, collect reproductive material. Do not collect plants in sterile condition. The only exception to this being some trees and shrubs that are recognizable based on their leaves. You can always tell what species a maple or an oak is by its leaves. So,
what if you've got a really rare plant? I mean, I really feel bad about taking out a reproductive plant. I mean, I guess if it's really rare, you wouldn't pick it up. But let's say it's on the edge. You've got enough of a population you can sample it. But if you're taking out one, not everyone's flowering. Maybe half, 1/10 of the plants are flowering, and 1/2. I know what you're saying. Lots of plants you need the flower to identify. But let's say it could identify without the flower.
Then do a reproductive, but take a picture.
Take a picture.
Of something else in the population that's in flower.
Okay.
So we've got that documented. For herbaceous plants, please collect the whole plant, roots and all. Don't just rip up the above ground part, but rip it out of the ground, roots and all. Collect at least one plant, and as I said, if they're small plants, collect more than one. So this is one of Robert Burton's specimens, which is a very nice layout of Canada Mayflower. You should also be thinking as you collect that herbarium specimens also have aesthetic value, that they ought to look pretty. And it's something I always think of when I'm mounting a specimen, how to arrange it on the herbarium sheet so that it is pleasing. And obviously, one single plant would serve to document the position, the place, but more gives you more of a range of variation and it just looks better. if you've got enough plants. Make sure you've collected sufficient material to identify your specimen. Remember what parts are important to collect to identify it, and make sure you have those important parts. Remember that with sedges and rushes, you've got to have mature fruit in order to identify it. A rush or a sedge, an Eleocharis or a Carex that's collected in anthesis, in flower, is useless because you can't identify it with any definitive certainty. You've got to have those mature fruits. So be aware of what you need to identify things and make sure you get that important structure. And as I said, try roots and rhizomes wherever possible. Of course, you will run into the situation where even an herbaceous plant, you've got a woolly mullein for Vascum thapsus, and it's 6 feet tall. That doesn't work. So what we recommend is collect a small part of it, or maybe 2 small parts. If I've got a 6-foot plant, I might collect part of the inflorescence and the base of the plant, showing its basal leaves. Some people would collect the whole plant and divide it up among 3 different sheets. So you've documented the whole plant on 3 separate sheets. For woody plants, get enough material, obviously, for identification purposes. Don't ever collect a single leaf. Get a leaf plus a part of the branch. So you've got buds, you've got twigs, at least. Some people even collect chips of bark and put that bark in with their specimens. The same thing about single leaves also applies to ferns. So many people, when they collect ferns, they collect a single frond. Well, that's a single leaf. It's great. You really want to have the whole plant, which means you want the rhizome as well. So we always try with ferns to get at least a piece of the rhizome or the crown so that we've got the whole thing. So how do you collect? With herbaceous plants, you go in and dig them up. In sandy, loose soil, you can pull them out of the ground, but generally you want to dig them up. And this is really important. Shake the dirt off the roots or wash the dirt off the roots. You do not want the person who has to mount that plant to have to clean the roots. So clean the roots. And if they're very large, cut them to shape woody plants. Whenever you collect your woody plant, that's why you carry your clippers. You want a clean cut. Don't go up to a woody plant and just rip the branch off, but do a nice clean cut, maybe so you can see the pith. And what I really ask people to do is collect extra bits. See, this is what we call a fragment folder or a packet. All of the NEBC specimens have a fragment folder on them. And traditionally that's used for the bits that over the years fall off. Because herbarium specimens, they're handled and used, bits break off, they fall off, and you stuff them into the envelope. But these days, people who are doing research, particularly phylogenetic research, want to do DNA analysis. So, like every week or so, I see a request for someone to take leaf tissue off of an older herbarium specimen, which damages the specimen. That's why we call it destructive sampling. So if, when you collect an extra inflorescence or an extra couple of leaves and press them with your specimen, when we mount it, they go in the packet. And when somebody wants to do DNA analysis, the stuff's right there. So if I was collecting this prunus, I would clap off an extra inflorescence and probably 2 extra leaves and press them so they could be put in the packet. So, yeah.
I was taught that, to press it with the dirt and it'll flake off when it's dry.
It slows. When I'm mounting plants, I do most of the mounting for the herbarium, and I come up with a plant where nobody has ever cleaned the roots. I have to stop my mounting process and clean the roots. And particularly for like mud, it's bad enough sand you can shake off, but if it's in a muck or mud substrate, you can't ever get that off unless you rinse the roots. And I even spray them with a hose. So that has to be done right away.
Okay, so you do actually wash those
Yes,
off? Okay.
if they need it. So we'll talk about collecting methods. We talked about field press, which I love to use field presses, but some people in the field, and sometimes I do, use different techniques. There is the plastic bag technique. Some people who are collecting carry a suite of Ziploc bags with them, or even large trash bags, depending on what they're collecting. And as they collect stuff, they just drop it into the plastic bags. I do this with grasses and sedges, but I have a slightly different technique. These are the plastic bags that my daily newspaper comes in. They are the perfect size for a specimen of a grass or sedge. You just drop one in, and there you go. They work really well for graminoids, but if you've got anything that has petals, if you put it in a plastic bag and then carry it for a couple hours, the petals will fall off. And you don't want that on your to have it happen to your specimen. So they don't work well for specimens, for things with flowers. They work fine for graminoids. They may work fine for ferns and for tree branches sometimes. But if you're looking at anything with any kind of flower, not a good idea. This is the old-fashioned field press. This is called a vasculum. These are metal, elliptical-sided metal boxes that the old-timers used to use back before we had plastic bags. They use those. And some people now, a friend of mine collects in a cardboard box. He walks through the woods and he just drops his plant specimens into this open cardboard box. And they don't get crushed, but they wilt. So there's nothing to prevent them from wilting. And again, if you're out in the summer for a couple of hours, things can get very badly wilted. So for those reasons, I really prefer to use a field press if I'm collecting anything other than graminoids, using a field press and pressing directly into my field press. And then field press, it's easy. You've got the newspaper right there. You label the newspaper. Everything's great. So another thing to think about when you're collecting your plant, particularly if it's a woody plant or anything that you're looking at, is the amount of real estate that you have to deal with. And here we go. I did a mock-up of this. Now, this is a standard herbarium sheet. It's about 17 by 11, 17 by 12. And some people, when they collect, and this is the size of your newspaper, they fill that sheet with plant material. But that doesn't work, because on that sheet, you've got to have a label. Labels are usually about 3 inches by 5. Above that label, you've got to have space for future annotations. If people change the name or correct the ID, you need space for that. The fragment folder has to have a space. The barcode has to have a space. The top corner, one or the other, gets the herbarium's identification stamp, NEBS or GH or UMass. So when you put all together, this dashed line defines your space for your specimen. And it's more like about 13 by 10, not 17 by 12. And that's something to think about. Don't have your specimen, when you press it, fill the whole piece of newspaper, because that will not work. Now we're getting to it. How do you collect a plant? You've got your plant and your newspaper. You've written on your newspaper what's in that. You open your newspaper. You place your plant carefully on the newspaper, fold it over, and put it into your press. If I've got a field press, it just goes into a pile of these. And I tend to put about 5 specimens and a piece of cardboard and 5 specimens and a piece of cardboard. The trick is how you arrange that plant, because you'll almost always have to fold it to put it on the sheet. And you want to fold it neatly. You want to fold it so that you've got leaves, so you can see the upside and the underside of the leaf, because both are helpful. You want to press flowers so that they're open. Many people press, and you press a flower so it flattens sideways. Well, if you do that, you're not going to be able to see the stamens or the anthers. You want to press a flower open. If you've got something in the aster family, say a daisy, you want to press one head up so you can see the flowers, and one head down so you can see the bracts. So think about how to arrange things. And you want to show diagnostic features. Are sheaths important? Make sure you can see a sheath. If it's, a ligule is important, make sure you can see it. Thick, large specimens might need special techniques. Like you can see this Angelica. They have taken the sheath and split it open so you can really see the whole sheath. They've cut this plant in several parts. This is a very thick stem. If you even cut that stem in half, you can see the structure of it. And that's sometimes really thick specimens require that kind of special technique. And I'm not going to talk about special techniques. Keith has promised to not ask me about this. Aquatic plants. I could do a whole class on how do you collect aquatic plants. It's a completely different animal. Very complicated, so we're not going to do it. Other really fleshy things like cacti are a whole special treatment. Thank goodness we don't have them here. At least only one. And even things that are really thick and fleshy. I collected water chestnut, Trappa natans, and that's really thick and big and thick and fleshy. And I sliced a lot of those in half so that they could be pressed nicely. But it takes a lot of practice to finesse your specimen, even when you're doing the quick and dirty field press, so that you've got things in the right order.
What do you do about cattails?
That's a special case, but I can tell you about cattails. It's a quick special case. So cattails, they're very thick, cylindrical. They can be as much as almost an inch thick. And you could, many people do, just press them as is. But have you tried to key out a cattail? The diagnostic feature for cattail species, especially for the cattail hybrid, is a detail of the base of the flower. You can't see that from the outside. So if you've pressed a cattail, a whole cattail spadix, you can't see the diagnostic features. So when I collect cattails, I slice that in half. And I press it one half up and one half down so that you can see the important characters. And how you can do this is you press into a field press, and that's your first go-round at doing this. You take your field press back home or to your car, and you transfer from your field press into this press. And then you can tweak things again, spread them out, turn that leaf over. Sometimes I'll even open up a press a day later, because after they've been in the press for a day, they've started to wilt, and they're easier to manipulate. Things might bend where they might snap if they're really fresh. So you need to tweak them. Sometimes I'll do that over 2 days. And then you just take your press and you let it dry. If you're lucky, you've got a drying room or a drying cabinet that has convective heat, so that you get these plant presses have corrugated cardboard, and that lets air move through between the specimens, which helps with the drying. For most of our NEBC work, I take our specimens into the drying room at Harvard and dry them there. In my personal stuff, I put in the warmest place I can find in my house. What you can also do is you can build a box. If you're doing a lot of this, you can actually build a box with light bulbs in the bottom of it and put your plant press like that across the top of the box. And the hot air from the light bulbs comes up through it. So there's several. Incandescent light bulbs, yes. Don't try this with LEDs. You don't get a lot of heat out of an LED. And check it, and it should be dry in a week. And the reason you try to dry them hot is so that they preserve their color and they don't go moldy. If they dry slowly, they could go moldy, and they could also lose their green color. So the fast you can. I've also been known, people don't recommend this, but I have occasionally turned on my oven, gotten it up to, 200 degrees or something, turned it off, and put my press in the turned off oven. Keith does it too. And then when it cools down, you might want to repeat this treatment. But that's kind of a kludge that we can do it. So you've got your plant, you've pressed it, you've dried it. What do you do next? Well, you got to type that label up. It's doing no good to you or anybody else if all you've got is a specimen in newspaper. So you want to take your data and type a label. And this has to be on acid-free paper, which you can get, like Staples sells thesis paper, which is archival quality acid paper. If you handwrite it, use permanent ink. I recently got a whole 100+ specimens where the collector had written out all the labels in pencil. I was able to actually copy them all on a copier rather than having to retype them, thank goodness. But you want to do this in permanent ink, 3x5 archival paper. And there's actually if you go poking around, there's a whole host of like computer programs on preparing herbarium labels. And you can even buy pre-printed or pre-perforated sheets that will go into your printer. So you might want to look for those. And then when you're done, if it's for your own collection, go ahead and mount it. If you're going to donate these to a herbarium, which we encourage you to do, most herbarium prefer to get them not mounted. So your herbarium would like to get the plants in newspaper with the label, and they will take care of the mounting. I don't care. I'm happy to accept things that people have mounted on their own. Keith brought me a specimen today. It saves me doing the work.
Shouldn't the specimen sheet be acid-free too?
Oh, absolutely.
Yeah,
Absolutely. But if you're going to mount it yourself, that's another whole workshop. There's a lot of tricks and how-tos about mounting herbarium specimens. You need herbarium mounting paper, which can only be gotten from a specialty source. You need acid-free glue, linen reinforcement tape. It's good to have fragment packets. So you need special material, and you really need to be taught how to do it. You can see it online, but you really want some hands-on experience. But it's another whole session. So I told you there are a number of references on our website. We've got our acquisition policy, which tells you what we're looking for in herbarium specimens, collecting guidelines, ethics, and a policy on permits and permissions. You can also learn a lot from other sources. There's a great paper that was just published in Rodora on specimen collecting, and that's available on Bio1 as an open access. I've got a handout for you on these. Open access on Bio1. And then if you just look at the internet, most of the major botanic gardens, Denver, Missouri, New York Botanic Garden, have really good videos on how to do plant collecting and pressing. And there are YouTube videos on this. If there's a YouTube video on everything, but there are good YouTube videos too. So, yes, and sources of materials. There aren't a lot of places to get materials. Herbarium Supply Company and university products are the best for papers and glues. Forestry Supply sells presses. I don't think they sell any of the other materials, but they do sell plant presses. And I think, no, these came from Herbarium Supply. So you might want to look around and see what you, if you want to start into this, see what you can get your best price. And they all sell like a starter kit, which is one press with blotters and cardboards. So, recap, what have we done? We've talked about collecting ethics, equipment, label data, really important, tips and techniques. And I would really like to do some practice. But I'll show you one. I brought in a lot of material, and there are some really nicely mounted specimens here if you want to see what the end product should look like. And one little tip that I didn't mention, which is handy for plants that have flowers, small flowers that you want to keep open, is what we call the spit technique. That you take a little bit of newspaper, you wet it, you pry the flower open, and you paste that piece of wet newspaper on top of it, and it holds it open. So
That's a good trick too.
So here I have a Norway maple, handsome plant. So we'll try to press it here. And as you can see, there's more of it than there is real estate on this sheet. It's a pretty good length, but I might actually cut that down a bit. But there are too many leaves here. And we could just bunch them all up like this and slap that on top, but that wouldn't be pretty. So I would sort of start from the top of this, and I'm kind of liking this for an arrangement. So I want to keep that. So I think I'm going to lose that leaf. I'm going to lose that leaf. That might be a good one to turn upside down. There's still kind of too many leaves on here. But, you know, that one, we could make that one go upside down. So, what do you think about that?
Good.
Kind of still a little crowded.
Where are you going to put the label?
We'll take the down leaf out.
Yeah, the down leaf comes off. Because with that in place, we would have no place for the label. Whereas this, we've got good leaves, we've got upside down, we've got a little bark here at the base, which is helpful. We've got some nodes and buds. And we can always throw an extra leaf in here to go into the fragment packet. So having done that, we can cover it over, put on a blotter, put on a piece of cardboard, scrunch it down. Let's do one more. Somebody get me a piece of newspaper. Oh, the next one's a bit of a challenge. I ripped this out of the ground, and I didn't take the dirt off the roots very well. Everybody knows what that is?
Is that garlic mustard?
Garlic mustard.
I've never seen one with pods that big.
I've never seen it that big.
It's a very
You always see
happy,
it in the spring.
Yeah.
it's a very happy garlic mustard. So there's a lot of garlic mustard there. We could probably do with just that much garlic mustard. So we will do the folding technique, trying not to go to the ends of the paper. And everything needs to fold. So we've got the fold, and we've got At least one leaf is right side up, one's upside down. That leaf could be a little better. Ah. So often it's helpful if you've got another person with you. So somebody can hold on to this and somebody else can slide the paper, yeah, paper and a blotter. No, paper first.
Oh, so we're gonna,
Slide that paper over and slap that blotter on it. Throw on a piece of cardboard. And if you were doing this just in the field with a field press, you would just do it in the newspaper and put up a stack of just the newspapers. But it's helpful to have a piece of cardboard with you to do that initial, let's squish it down first. And that's why I put a piece of cardboard between every, 4 or 5 sheets, because otherwise it's kind of a mess. But the nice thing about the herbarium supply field press, I'll show you, is that it's compressible. You put your specimen here, and you can actually make it tight. So it actually keeps pressure on your specimen.
And when they're in the main press, you need a cardboard sheet between each
Yes.
specimen to keep them from interacting with each other.
Yes, and also for the air
And the
circulation.
Air circulation, yeah.
So that's your basic of how you do it. And the specimens I brought in show you pretty nicely, folded. So that's a nicely folded specimen. Vines did a nice job of making that curl, so it looks good. And he's got, leaves are right side up and upside down. A little harder to do really dense shrubs like this, but, oh, and this is interesting. You can see it's got a bit of a spider web on it. It's actually nice to get plants that aren't perfect, that show evidence of insect herbivory or even insect use, because people actually will look for that. Somebody was studying herbivory using herbarium specimens recently. This got the rhizomes and everything, so we've got a nice representation of everything that ought to be there. A sedge. When people collect sedges, they often collect like a whole clump and press that whole clump. And this is actually perfect, because with the whole clump method technique, you often can't see the sheaths at the base because there's just too much stuff there. But this is separated out, and this is folded pretty well. You always have to fold them and crimp them. A ginger, nicely pressed with several individuals.
is that non-native?
It's non-native. This is the European ginger.
Neat.
And again, this, well, this one could be better. Wild geranium. But look at the flower. Flower is pressed sideways. He didn't press another flower in the open position. So you can see the petals and you can see the sepals on this one. But if you really wanted, if you had to look at the anthers, you couldn't find them. And that's really important with things with tubular flowers, where it's important to see like where the anthers insert on the tube. You want to be able to pull them out. Well, thank you all for coming. I hope this inspires you to get out there and collect, especially, collect for an institution, collect for NEBC, collect for UMass, collect for the Woods Hole Herbarium,